In this video, we're going to learn about mistakes in DNA replication and protein synthesis, which results in mutations. So we've already learned about some mutations that lead to different traits, such as skin color. Also, we learned about things that might be harmful mutations, like with some of the genetic disorders we were learning about, like sickle cell anemia or cystic fibrosis. So in this video, we're going to learn about both good and bad mutations, how they come about, their different types, and things like that. So the first thing is, what causes mutations? There are two main things that can cause mutations. One is mistakes in DNA replication. So if you remember back to our earlier unit, every time a cell divides and makes two daughter cells, each daughter cell needs a nucleus with the correct amount of DNA in order to function properly. So in DNA replication, the DNA is copied, so something called sister chromatids get made, and then those sister chromatids get divided into two cells. So the resulting cells are genetically identical to the original cells. But DNA polymerase, which is our DNA copying enzyme, makes a mistake about every 100,000 nucleotides. So maybe it's supposed to have a T and it puts in a G. Okay, so here you can see the mistake that might happen. So here it's trying to copy. We're trying to make two identical DNA strands. This one, it turned out great. The A and T are still together, but instead of A and T, we have A and G. So this is the mutation. And next time it gets copied, that G that wasn't supposed to be there gets passed on. Now, the cells do have mechanisms to repair mutations. So even though DNA polymerase makes mistakes, there is something called excision repair, where when there are these bumps in the DNA molecule because the bases aren't pairing right, enzymes can come in and cut out the broken part and put in the correct nucleotides. So the body does try to fix itself and tries to repair mutations, but sometimes they do get missed. Another thing that can cause mutations, mainly because it damages DNA, are environmental factors. And there are three main groups of environmental factors. The first group is radiation. So some factors, such as radiation, actually physically damage the DNA, and they make it hard for DNA polymerase to correctly copy it. So here you can see, here's an incoming UV photon. And what happens is, instead of these yellows are supposed to represent thymines, instead of thymines binding to adenine across the way, UV light causes these things called thymine dimers. And basically what happens is the thymines bind to the other thymine rather than the A's across the way, and you get these bumps. So let's say this is our DNA reading machinery. So maybe it's RNA polymerase if we're trying to make a protein, or DNA polymerase if we're trying to copy the DNA. And it goes along and it hits that bump, and then it falls off. So basically our enzymes don't work if our DNA is damaged and has these bumps. And so you can't make proteins from it, you can't copy it correctly, and that can be very harmful to the cell. Other factors that can also cause damage are things like different chemicals, like carcinogens, preservatives in foods, chemicals in cosmetics and cleaning products and things like that. All of those can also physically damage the DNA, or they can damage the cells. So for example, cigarette smoke damages your cells. When you're breathing in chemicals, either from smoking a cigarette or through vaping, those chemicals damage the cells. And then the body has to make new cells to replace the damaged cells. And every time the cell has to copy its DNA, there are always a few mistakes that are made. Okay, right? Because DNA polymerase makes mistakes every 100,000 nucleotides. So if you are breathing in lots of cigarette smoke, or maybe you have a job where you're breathing in asbestos, or things like that, you're constantly damaging the cells of your lungs, and so your body's constantly having to repair. And when you have to repair a lot, there's a chance that those new cells might not be repaired correctly. Another thing that can happen, and we learned about HPV earlier in the year when we learned about cervical cancer, but sometimes infectious agents like viruses can lead to mutations, which can lead to then cancer. So viruses inject their DNA into their cells. If that DNA lands up in the middle of a gene, it could change the code of the gene, so now it can't make the protein. So we saw that HPV can take you one step closer, for example, to developing cancer because maybe it shuts off a tumor suppressor gene. Or maybe it messes up a proto-oncogene so it goes super fast. So lots of different environmental factors can lead to mutations. So we can't prevent mistakes in DNA replication. That just is a natural thing that happens. But we can prevent our exposure to radiation, chemicals, and we can get, say, vaccines to prevent against different infectious agents to reduce our risk of developing mutations due to environmental factors. So let's take a look at types of mutations. What we're going to focus on in this video are things called point mutations. So point mutation changes the DNA at a point in the DNA sequence. So it changes one or a small number of nucleotides in the DNA sequence. So it's changing it at a point. And there are two main types, substitutions and frameshift mutations. So this is our normal sequence we're going to be using as an example. Okay, you can see it has 31 codons, which means it's going to have 31 amino acids. And what this protein is coding for is this is going to be part of melanin, which is our protein that causes skin coloration. So we learned about this with our previous case study, right? Melanin helps block UV light, protecting you from UV damage that can lead to mutations like we see here. So it's good to have more melanin if you live, say, closer to the equator where you have more UV exposure. All right, so our first type of point mutation is a substitution. So a substitution is when one base, so one nucleotide, is substituted for another. So substitutions can vary. There are three different types. There are these things called missense, nonsense, and silent mutations. And they get their name based on how do those mutations affect the proteins involved being made. Okay, so we have two different types shown here. The first one is a missense mutation. Okay, so here is the mutation. We're supposed to have a G, but we have substituted in a C. And what that means is you're now going to get a different amino acid. And as a result, the protein has a different shape. So let's go back and look at the normal protein, right? It has these two, what we call disulfide bridges that are holding it together. But when you have this mutation, we're missing that second thing. So it's not holding it together into the same 3D shape. So what that means is we're going to get a different shape. And so if you get a different shape, it means you're going to get a different function. Shape determines function. That's a key idea in biology. So we call this a missense mutation or a missense substitution because our protein doesn't make sense. So in this case, this abnormal pigment isn't going to produce the same melanin, and as a result, it won't have a dark skin color to prevent against UV radiation. This person with this mutation would have lighter skin color. Another example of a substitution is a nonsense mutation, and that's down here. So we were supposed to have a T, but now we have an A, and what that means is you end up with an early stop codon, okay? So basically, in this case, a nonsense substitution, you end up with a short protein. So it's not as long as it normally is. It's only 19 amino acids long, whereas it's supposed to be 31 amino acids long. So we have a short protein. And again, we're going to have a different function because we have a different shape. So you can see, because it has this different shape and it can't function, this protein is complete nonsense. It doesn't work at all. So again, in this case, melanin isn't made properly. This person's going to have light skin. Now, the last type of substitution is something called a silent mutation. A silent mutation, there's still a mutation. So here we have a C instead of a T. But what happens is we end up with the same amino acid. So there's no change to the amino acid sequence. So what that means is, even though there's a mutation, it doesn't affect the protein at all. And the reason is because in our genetic code, we see that there are multiple codons that code for the same amino acid. So our codon is UGC, which is this right here. So you can see there are multiple codons that still code for it. So by chance, okay, that T became a C, okay, and now we have kind of the same amino acid being made, and we have the same protein. So in this case, this melanin has the right shape. You get the normal skin tone. You get the nice brown color of your skin to protect against UV light. Let's look at a real-world example besides skin color of an substitution that could be potentially harmful to an individual, right? With skin color, we learned that your skin color could be beneficial depending on where you live in the world, right? Lighter skin is beneficial closer to the poles because there's less UV light, and you don't want such dark skin to block your ability to be able to make vitamin D. Whereas closer you live to the equator, the more beneficial it is to have dark skin tone so that you can prevent cancer-causing mutations from UV damage. So there's definitely benefits to both types of skin tone depending on where you live. Now, sickle cell anemia doesn't really have any benefits. The actual disease doesn't. We can talk a little bit about evolutionary why it's still around in a minute. But basically what happens is we have a missense mutation. Here is part of the normal hemoglobin protein. And sickle cell anemia is typically caused by one substitution. So there's a valine when there should be a glutamine. Okay? And normally hemoglobin is these individual little proteins that float around in our red blood cells, and our red blood cells look almost like little dimpled donuts. Okay? However, because of the valine in the mutated form, okay, valine is hydrophobic. It doesn't like water, so the hemoglobins don't want to be in water, and they stick together and they form these chains. And these chains stretch out the red blood cells, so they're long and skinny. And what happens is when you have a blood vessel, those long, skinny red blood cells can get stuck, and that can lead to heart attacks and strokes and things like that. Now, sickle cell anemia tends to be found in places, in populations that live closer to the equator, because although the actual sickle cell anemia is negative, being heterozygous for sickle cell anemia. So, for example, being like big S, little s, you have, it's an example of codominance. You see both. So, you see some normal ones, but you also see some sickle cell ones. Not enough to cause a lot of these blood clots, but enough to prevent malaria infection. So mosquitoes can carry the malaria parasite, and malaria parasites love round blood cells. They infect it, and then when they reproduce, basically they burst out of the cells and they kill the red blood cells. And that can lead to death over time. But malaria has a really hard time infecting these sickle cells. So if you're heterozygous, you get the best of both worlds. You have the dominant allele, which means you still get some normal red blood cells. You don't have blood clots, but you have some of the abnormal cells, and that allows you to avoid a widespread malaria infection. So you don't have sickle cell anemia, you don't get malaria, you survive and reproduce better. So again, mutations come across, sometimes they're bad, sometimes they're good. It's really, you need to look at what are your conditions in your environment. All right, so let's learn about the other type of mutations, which are frame shift mutations. So we had substitutions, now we have frame shifts. So frame shifts come in one of two forms. They're either insertions or deletions. And what happens is when you add or take away a nucleotide, okay, all of the bases after the point of the mutation are changed. So that means every codon after that point is changed. Okay, so what that means is you're going to end up with lots of different amino acids. So you're going to get a different shape, which means a different function. Okay. All right. So if we go back and we remember, go back. Here's our normal melanin. Remember, it has those 2 disulfide bridges. It's 31 nucleotides long. Okay, let's look at this frame shift mutation. Okay, in this case, first thing we're looking at is an insertion. So we're adding a nucleotide. We're adding a G. And now every codon, okay, is getting shifted over. So all of these amino acids are different. That's why they're colored in white. So they're all different from the original. And this keeps going on and on. Okay, so you can see we have a totally different list, which means our amino acids are going to interact differently. We're not going to get that second disulfide bridge, and it's going to fold into a different shape. In this case, the new shape makes the pigment actually darker, and so this individual has really dark skin. Okay, so again, this is a different mutation, and again, this might be positive if you live in, say, closer to the equator where there's a lot of UV light. Or it could be detrimental if you live closer to the poles and you have an inability to make vitamin D as a result. All right, the other type of frame shift is a deletion. So deletion, you remove a nucleotide. So in this case, we took out a G. So again, now all the codons are different, and we're going to code for lots of different amino acids. So it's just showing here, but after this point, they're all different. And it may not even stop at 31. It may keep going until you hit a stop codon. So you're going to get a very different protein. Because this happened earlier on, we can see that there's not even that original disulfide bridge that was holding it together. So now it has, again, a very different shape. So it's going to have a different function. We're basically completing a new protein. So they're basically saying this could lead to a nonsense, the protein making no sense whatsoever. So it's really problematic to have an insertion or deletion because they cause big changes to the proteins. So substitutions, okay, are just changing one nucleotide, which could change one amino acid. and that could change the protein, but it may be for better, it may be for worse, or it may be silent and not change the protein at all. Frame shift mutations, on the other hand, because they're not just changing one amino acid, they're changing lots of amino acids because all of the codons get shifted, you're going to see a very big change to the protein shape and therefore its function. So let's look at a deletion example. Cystic fibrosis is caused by mutated salt channels. So this is a normal salt channel, and chloride ions normally are able to pass in and out of the cell, and that keeps the cell from becoming too salty. Okay, if the cell was too salty, water would flow into the cell, and that could be harmful to the cell. Okay, so the protein is called the CFTR protein, and the mutated protein basically shuts down the flow, and it blocks it so that no chloride ions can flow out. And that happens because 3 nucleotides get removed. So we deleted essentially a phenylalanine. So rather than just deleting 1 nucleotide, we had 3 nucleotides, which means we essentially delete a whole codon. Now with 1 amino acid missing, it changes its shape. So let's look at normal versus mutation. So normal, salt goes out, water goes out, and you end up with thin, watery mucus in your lungs. That mucus traps bacteria. It allows the cilia, the little hair-like structures in your lungs, to kind of sweep away that bacteria and get rid of any infection you might have. But if you can't get salt out of your cells, water basically isn't going out to dilute the mucus. So you get this really thick mucus. And so as a result, these little hair-like structures, those cilia, they get basically gummed down. They get stuck in the mucus, and they can't sweep away bad bacteria. And so you get all this bacteria growing. And people with cystic fibrosis tend to die early of lung infections like pneumonia and things like that. So here you can see all this bacteria growing in this thick mucus because they can't sweep it away. So here's our mutated protein. Okay, so if cystic fibrosis isn't treated, people typically die by the age of 5. Now with treatments, people usually live 30s or middle age before they may succumb to a lung infection like pneumonia. So we have treatments to extend their life. Scientists are looking to use gene therapy to try to replace the mutated gene with a working gene to basically provide a forever fix for people with cystic fibrosis. They are also looking at using something called CRISPR, which is a gene editing tool to try to fix the mutation to help, again, heal the gene so it can make working proteins. We'll learn more about that in our next unit. So, our key idea: mutations are harmful when they change the shape of the protein produced so it can't function in the same way. So remember, DNA determines messenger RNA makes your proteins, and proteins determine your trait. It's the shape of the protein that determines its function. So here we have this enzyme amylase. Okay, amylase you see has the right active site to allow starch to come in and for starch to be digested into these mini sugars called maltose. So basically it's something you have to do to try to digest the starch in your food so you can get energy from it. If your enzyme is mutated and it has the wrong active site, starch can't fit. Okay, starch would build up, you wouldn't be able to get energy from it, and you'd have less energy. So shape determines function. It's really important that proteins have the right shape to do the right jobs. So what we just talked about was a loss of function mutation. So this is a bad mutation. It could potentially harm or kill an individual with it. But not all mutations are harmful. Okay? We talked about silent mutations. silent mutations have no change to the protein. Even though there's a mutation, the same amino acid got coded for, that person is still fully functioning, has normal proteins. Sometimes we have a gain of function mutation, okay? So where the protein works even better than before, and now they can survive and reproduce better. We are the result of lots of gain of function mutations, okay? Our ancestors had mutations, and those with bad mutations died out, and those with good mutations were able to survive and reproduce and pass those mutations on. So we are who we are because of the good mutations in our past. Also, some mutations can sometimes occur in non-coding areas of our chromosomes, so things that are not genes. So they have no effect, and we call these neutral mutations. So for example, introns that get cut out, if you have a mutation in an intron, it's going to get cut out and it never makes it into the protein, so no big deal. So sometimes mutations just happen in places that don't make proteins, so they don't determine our traits. We talked about skin color being a beneficial mutation, but here's another example of a beneficial mutation, and it's tolerance to high cholesterol. So every person has this apolipoprotein, and this protein basically traps cholesterol in our blood. If we do not have a lot of this protein, or it doesn't work very well, or we eat too much cholesterol, so our protein can't collect it all, we end up with these cholesterol plaques in our blood vessels, and it makes it really hard for blood to go through, and that can lead to heart attacks, strokes, aneurysm, things like that. So basically blocked blood flow. Now, there was this small town in Italy, and scientists found, when they were studying them, that these people in Italy could consume cholesterol-rich foods and show no sign of heart disease. So no sign of strokes, heart attacks, any of this cholesterol buildup. Basically, this whole stuff was missing. There were no plaques, even though they ate an incredibly cholesterol-rich diet full of like butter and cheese and red meat and things like that had lots of cholesterol. Okay, and they did genetic testing, and they found that this population had a mutation that produces an apolipoprotein that works even better than normal. So basically, it gets all the cholesterol, and it removes all the excess cholesterol from your body, so you never get plaques. And so basically, they developed an immunity to heart disease. And because this town was basically isolated in the mountains, everybody kind of reproduced together. And so this mutation was passed from generation to generation, and it became really common in this little small town. And so there's this town that can survive heart disease. It's pretty cool. So the last thing we need to talk about is something we've mentioned before, but it's just important to remember that all mutations are not inherited, okay? So mutations in body cells are not going to be inherited. So for example, let's say Jenny likes to go tanning, and she goes to tanning beds, and as a result, she has damage, UV damage to her cells, and her cells have become cancerous. Because she develops skin cancer, and her skin cancer is in her skin, her children are not automatically going to be born with cancer. You don't pass your skin to your offspring. Only mutations in eggs and sperm are going to be passed on to the next generation, okay? Because eggs and sperm are what contribute to the offspring. So in this case, that town in Italy, the only reason they were able to see this mutation happen over and over again and be passed from generation to generation is because it happened in the original person's. It was a man they traced it back to, his sperm, and he was able to pass it on to his kids, who were able to pass it on to their kids. And so that is how it was able to be passed from generation to generation. So hopefully this helps you understand mutations, how Mutations can be both beneficial or harmful depending on the scenario and how the protein folds. So just remember your key idea: structure relates to function. You change your amino acid sequence, you can change your structure, and that could change the function for better or for worse.